what's in them, in this cell, they're like miniature organs. So the organelle. And they have functions, right? And so when you take all the organelles together, each has a particular function, and that's going to allow the cell to live. So a cell is the smallest living thing. So I'm gonna want you to know what the organelles do. So I'm gonna list about here. I'm gonna list the organelles, and you only need to know just one word would probably be good. 1 or 2 words would let you know what they do. So the organelles in here, so this is like an RV, right? And then RVs have an electrical system, they have a toilet, they have lights, they have a stove. That's like this. So it's self-contained. So I'm going to start with ribosomes. And ribosomes make proteins. I can see that. Hold on, let me just make a slight change. So ribosomes make proteins. So in here you've got ribosomes. You have this thing called the endoplasmic reticulum. I'm just going to put ER, so don't even bother learning the word. There's 2 types of ER. Some of this ER has ribosomes on it. We call that rough. And because of that, it makes proteins. And then there's also some of this ER that does not have ribosomes on it, and it's smooth. It makes lipids. So a cell can make its own proteins, ribosomes, rough ER, they both make proteins. And it can make its own lipids. And then we have the Golgi, and I'm putting just Golgi because it's called different words. Golgi apparatus, Golgi complex, Golgi whatever. So Golgi makes its own carbs. And more importantly, it takes the proteins and it packages them and it repackages them and sends them out. It's like if you're working at Amazon and you're sending stuff out, right? You're not going to pack this in like a super long, skinny box, right? You put it in the appropriate box. And so that's kind of what the Golgi does. The next 2 we've already talked about. Mitochondria. Mitochondrion is singular, mitochondria is plural. It's not important whether you know which is which, just mitochondria. This is where ATP is made. Remember, this is what's behind cellular respiration. When we talk about cellular respiration, we're talking about what's going on, well, in the cytosol a little bit, but mostly in the mitochondria. And then the next one is centrosomes or centrioles. I'm using them interchangeably, centrosome, centriole. That's involved in mitosis. So these things that I'm writing next to it, if you know that, you'll get all the questions. And I'm going to give you one more, and that's not all, that's not really everything. So I'm going to give you one more. It's a lysosome. Lysosomes have digestive enzymes. So these things are common to animal cells. If we were talking about plant cells, there would be a couple of small differences. For example, plant cells have chloroplasts, but we don't have plant cells on this makeup, so I'm not going to go into plant cells. The other thing I want you to know is that this is a eukaryote. So there's these two types of cells. I'm going to write it here. There's cells called eukaryote, and there's a cell called a prokaryote. The difference between them is very easy. Prokaryotes are bacteria. Everything else is a eukaryote. Bacteria are prokaryotes, and then there's like this other type of cell called archaea, which you're not going to really see. So practically speaking, bacteria are prokaryotes. Everything else is a eukaryote. So that's it. Just ask yourself, is it a bacteria? Yes or no? That's it. Yes, it's a prokaryote. No? Then whatever it is, it's a eukaryote. Like our cells, like this cell that I'm talking about. So sometimes the way that a bacteria does things is different than the way that other cells do things. And so those are the things that are inside. Here's the nucleus, and then you have all these organelles, however you want to however your book does them. I don't know what they are. Then there's this ER right here. So it looks, a cell looks something like that. It's got a bunch of stuff in it. All the stuff that's in it, that's these organelles. These organelles are floating in cytosol. Together, when I take everything that's out here, the liquid and the organelles, I can call it cytoplasm. But, yeah, so that's the cells. I mean, what would I ask you? I'm going to ask you something. I'll ask you something from here, or maybe I'll just say, talk about the organelles you know. List 3 organelles, list 5 organelles, and what do they do? Just the word is fine. Mitochondria, ATP. I'm not looking for exact spelling, even though I should, but I'm not. So, but you got to get it. You have to get it so a reasonable person would look at the word and say, "Oh yeah, she's trying to say lysosome is just spelled wrong." That's fine. You can't put L, scribble, completely wrong words. Oh, I knew the word started with L. No, you should be able to tell me, if I say, oh, what word were you trying to write? And you'd be like, lysosome. Oh, okay. Or this word, like mitochondria, right? That can easily get spelled. That could go really wrong, right? But if I were to ask you, you'd be like, oh, that's mitochondria. So don't kill yourself on the spelling. This is just memorization. So this is rather easy. And I would go on, there's lots of websites that you can go on to learn the parts of a cell. And just Google parts of a cell. There's interactive websites that are really good on it. Remember that animal cells and plant cells and plant cells are eukaryotic cells. And only bacteria, they are prokaryotic cells. Bacteria are much simpler. So that's it on cells. We are going to revisit, when we talk about cell cycle, we're going to talk about centrioles, centrosomes, and then mitochondria when we go over respiration. I definitely want you to know what this membrane, I know you don't get put off, I'm not looking at you, but I'm just like, this membrane, you need to know what it's made of. It's called cell membrane or plasma membrane, and it is a It's made from 2 layers of phospholipids. I asked a few questions on this chapter because I thought it would be easy, and a lot of you it didn't. It wasn't easy. First of all, if you're taking multiple choice tests in the future for any class, don't focus on what you don't know. That teacher might be making up words. And so you're seeing, like, you know A, you recognize B, C and D are like these complex words, and you're like, and then you start freaking out. Oh, that's just more shit I don't know. They might have just made those words up because they didn't know what to put for C and D. Don't focus on what you don't know. That just whatever you know is the one that's there. Or they'll be like, which one is not present in cells? And you get all these words you've never seen. The thing that is not there, like I'll be like, what's not in animal cells? And I give you all these words that you've never seen in your life. But one of the words is chloroplast. Chloroplast is in plant cells, right? So you recognize, you're like, oh, chloroplast. Yeah, that's it. I don't know what those other 3 words are. Don't worry about it. So, anyway. But we're not doing multiple choice. Okay, so phospholipid bilayer, you need to know that. And so this chapter here on the cell membrane is talking about this phospholipid bilayer. This cell, when it makes stuff, it needs to put it out of the cell, and it needs to bring stuff into the cell. By the way, when the cell makes things and kicks things out, what do we call that? That was one of the questions. Cyto means cell. Exo means out or outside. Right, so that's when a cell puts things out. The cell is also bringing things in, right? The cell's got to eat. The cell's this is a living thing. And it has to get, it needs water and it needs sugar. It needs some kind of food. And then it's got to get rid of all its shit, so it's got to like put it out, right? So it's taking things in and it's putting things out, just like you do. So putting things out, exocytosis. Bringing things in, endo. If you remember, there was different types of endocytosis, and you just need to ask yourself, eating, drinking, or bringing in something that's very special, a particular thing. Eating, phagocytosis. Phagocytosis. I guess I'm going to write the whole thing out. Drinking, pinocytosis. The third one, the one that's very specific when you're bringing in one type of thing and not just anything, receptor mediated. That's the type of question I would ask you on a test. That's a possible question. I would say something like, describe the types of endocytosis. Then you're going to be phagocytosis, pinocytosis, receptor-mediated cytosis. And then you're like, oh man, these are all hard words. Okay. This word endocyte, it's the same word. Cytosis, cytosis, cytosis. You're just memorizing the beginning words. This is cell eating. So if you know how to spell the word esophagus, you remember eating esophagus, then you're remembering ESOPHAG. There's my word. Pinocytosis, I don't know. pina colada, whatever helps you remember it. And then you know that one's drinking. You've seen both of these words before, it's just now we're combining them. Receptor-mediated endocytosis. And then what are they? You could use one word and that would answer it. I would be okay with that. Eat, drink, specific, or special. Or if you want to use more than one word, not just anything. That all answers the question. So definitely know this. Okay, so bringing things in, bringing things out. How does it, if I only give you one choice, how does this happen? There's more than one way it happens. There's many ways it happens, but you only get to choose just one thing. So how does it happen? Just give me one word, and you think it to yourself. What's that one word? How do things move around the body? You just get one word. I hope your one word was diffusion. It's not the only way things get around the body, but it's the main way, diffusion. On that test, the key words you should have been looking for with those questions, when I talked about passive transport or active transport, you should have been looking for high and low. All the other words in that sentence didn't really matter. Higher to lower. If it was active transport, you were looking for lower to higher. So diffusion is the movement of something from higher concentration to lower concentration. I will ask you about this. It's the movement of oxygen, carbon dioxide, water, calcium, sugar, whatever it is. It goes from higher to lower concentration. Your body is all equaled out, all equilibrium. Got the same stuff inside as outside. It likes to keep the concentrations of stuff equal on both sides. Nice balanced. Your body tries to balance itself all the time. And so that's diffusion. Things will go from a higher density or concentration or pressure, whatever the word is, from higher to lower. Another way to say this, what I just said, concentration gradient. Things follow their concentration gradient. Meaning if you got a whole bunch of oxygen around your lungs, it's going to start flowing through your body until it reaches the other places in your body. That's diffusion. Following the concentration gradient is the same thing. So if I were to say something moves against its concentration gradient, that's the same thing as saying against diffusion. So what's the opposite of this? Lower to higher. So this is concentration gradient. If I say against, it's just the opposite. Do you need energy to make this happen? No. This is passive. That's a passive process. It just happens. You don't need to give it energy. Erase myself. You don't need to give this energy. So you need to know the difference between passive and active transport. And I think I asked it on the test. I don't know, I'm gonna stop talking about the test. I have to move on. It's not just depressing for you guys. So, yeah, you have to know what is diffusion. So if I could just ask you, just what's diffusion? That's it, just like that. You write down what it is. What's the difference between passive and active transport? Passive, no energy, therefore active, energy. But maybe I want you to elaborate, like, well, tell me something else besides that, besides just energy and no energy. Well, okay, so active, we know active uses energy. Active is against concentration gradient or against diffusion. I would even take that word. This is how they're different. And really, if you know this, then you don't have to memorize this. This is just the opposite. So you could just memorize passive. Well, passive is no energy and it uses diffusion, which means that it goes from higher to lower. Okay, then what's active? Okay, what's the opposite? If I just said higher to lower, this must be lower to higher. If this is concentration gradient, this must be against concentration gradient. If this one's no energy, this one must be energy. They're just opposites. And remember, both of these ideas we talk about in cellular respiration. This is the idea behind the electron transport chain. So you have to know the difference between the two. It's likely I'll ask you about that. So that's another thing that you need to know that I could possibly ask you about. And I'll say discuss the difference between active and passive transport. So I'm using the word discuss. So, you just say this, energy, no energy, but then also, since I'm saying discuss, talk about how one is the movement from higher to lower, and then the other one's the opposite, movement from lower to high. Well, this is the concentration gradient is another way to say it. That's just another way to say the same thing. So far, okay? You get the idea? I'm going to erase this. Another thing that I want you to know about is osmosis. Which is simply the diffusion of water. Oh, and even before I start getting on this hyper and hypotonic thing, I forgot to tell you something. There's facilitated and regular diffusion, right? They're both diffusion. They're both going from higher to lower. But I might ask you to explain why would a cell use facilitated diffusion instead of just diffusion? So remember, facilitated diffusion is the gate. You're opening the gate. They're not just walking through the turnstile. So facilitated diffusion is still diffusion. It's still going from higher to lower. It's just you're helping it. You're facilitating it. That just means help. You're just helping it along. Certain things cannot just go into the cell and out of the cell. Anything with a charge. Anything that's plus or minus. Any cation, any anion. Things that are charged, they cannot just go in. They cannot just enter a cell or exit a cell. they have to have special gates that they go through. They're proteins. Anything that's too large, just physically too large to fit through, it's got to use a gate. Third one, you want to move lots of something at once. And I gave you the example of aquaporins. That moves a lot of water. That's just an example of moving something using facilitated diffusion. Water will go into your cell without an aquaporin. Water moves by simple diffusion. It goes into your cell. It goes in and it goes out. No problem. Just trickles in and trickles out. But if you want to move a lot of it quickly, then you got to use facilitated diffusion. You got to open a gate and let it rush out. So that would be like an aquaporin. All right. So osmosis. I want you to know the difference between hypertonic and hypotonic. It's just the amount of solute. What has more, and solute can mean just stuff that you mix with the water, right? So what has more solute, like coffee or a glass of water? Right? The coffee, the water is just water. The coffee's got something in it. It might have sugar, but it has coffee. It's something. There's something in it that's something that you dissolved, right? So the coffee would be hyper, because it's got more. The water doesn't have any. We call that hypo. I cannot take this coffee and say it's hypertonic by itself. I haven't compared it to anything, right? So these are words like whatever has more solute is hypertonic. Whatever has less, because you got to compare it, is hypotonic. And water, and this is about diffusion, water likes to follow solutes. So water works a little bit different. Water does diffuse. Water goes from higher concentration to lower concentration. But water is different than other things. It'll go from higher to lower unless it sees solute. Once it sees solute, it wants to go and follow it. So we have to give it like different rules. As far as water is concerned, when water sees solute, it thinks that that is the lower concentration. So you have a glass of water and you have a glass of salt water. A water molecule wants to go to the salt water. It feels that the salt water that's the higher to lower. Which doesn't really make sense for us, but that's just what it does. So water always moves from hypo to hyper. So you need to know that. Water always goes from hypotonic to hypertonic. And I gave you some question about celery stalks, blah, blah, blah. You're taking two things. You were supposed to take celery stalk and water. Now, whatever that liquid is inside the celery, that's hypertonic. Because it's not just water, it's water with whatever stuff is in the celery stalk, right? So, and then the water was hypo. So when you figure those two out, then you know what's going to happen. So I've got a celery stalk, I've got a glass of water, I put the celery in the water. The water's hypotonic, the celery is hypertonic. It's got stuff in it, whatever it is. So I already know where the water is going to go. It's going to move into the celery. Do you guys have a class? Do you have a class coming up?
Yeah,
Durga, Andrew? At 10?
yeah.
Okay. Thank you. All right. I'm gonna have to drop off for a few minutes. Let me finish this thought, then I gotta drop off for about 3 minutes. I'm gonna go open my laptop and I'm going to jump back on, because I gotta abandon this classroom. So you just think, what has more solute? That's the direction that water is going to flow. So you need to know that, hypo versus hypertonic. It's very likely I will ask you about that. So on this chapter, membrane, we have hypertonic versus hypotonic. We have different types of endocytosis. We have active versus passive transport. Why would you use active? Because you're going against a concentration gradient. You have the two different types of passive transport, right? So there's like 4 things there that I would ask you. I'll pick a couple of them and ask you a question about it. And they'll just be kind of open-ended questions. So that's everything on the cell. That's everything on both the cell and the cell membrane. I'm going to stop recording. I'm going to go run somewhere in about 5 minutes. We'll take a 5-minute break. I'll try to go to another classroom. And if I can't, I will just open my laptop. So you don't even have to get off the meeting if you don't want. Just take a 5-minute break.